In this video, we are going to talk about digestion system in human bodies along with the alimentary canal is associated glands along with the organs. So let's start it. When we think about digestion in human bodies, there are primarily three molecules which we are concerned with. It is the starch molecules, the fats, and third is the proteins. Okay, let's start off with the mouth here. So when we consume any food, mouth is essentially the gateway through which food is entering into a human bodies. So mouth is just a transverse slit which is bounded by two soft movable lips. But these lips are having a special importance in controlling the regulating the opening and closing of our mouth, but along with helps in controlling the finer precisions of our speech as well. After entering into the mouth, the food then reaches the buccal cavity. The buccal cavity has essentially three main organs. First is the teeth, second is tongue, and third is the salivary glands. When we say about teeth, it helps in mastication, which is essentially chewing of food or crushing the food into small tiny particles which are helping in increasing the surface area for various digestive enzymes to act upon it. Afterwards, the tongue, it's the organ of taste and has numerous taste buds which are present on its surfaces, and it helps in detection of taste, and second helps in directing the food particles at specific spot in on your teeth, so helping in mastication in such. The salivary glands. So salivary glands are secreting the saliva. Now the saliva contains the enzyme called as the salivary amylase. The salivary amylase primarily helps in the digestion of starch. So for example, any forms of food molecules which is rich in starch is consumed by our human body. We are by the actions of the salivary amylase itself present in the mouth, it will get converted into simple sugars which is maltose and dextrins. Okay, these simple sugars are later further broken down into glucose which could be absorbed by the small intestine. But in such, the digestion of starch itself starts in our mouth itself, and later it's absorbed or digested further in our small intestine. Okay, so we can say starch digestion in human body essentially starts from the buccal cavity itself or in the mouth itself. Next, the food latter, the masticated or the food which is mixed with saliva is reaching into the esophagus. This esophagus is a common, it's a esophagus is connecting the buccal cavity along with the stomach. Its movement is currently guarded by the peristalsis. So peristalsis is essentially the rhythmic contraction and relaxation. Okay, peristalsis. Peristalsis is the rhythmic contraction and relaxation of the muscles of esophagus through which the food is circulated or passed from the mouth into the stomach. Now, when we speak about stomach, the stomach, it's the organ. It's a J-shaped organ, as you can see over here. Stomach, it's a muscular J-shaped organ which is situated in the upper left part of our abdomen, just below the diaphragm. So this yellow structure which I have drawn is the diaphragm. Okay. So diaphragm, below the diaphragm you have the stomach, which is a muscular J-shaped organ, and it's also regulated by the peristalsis movement and helps in churning or mixing the foods with the enzymes which is helping in converting food into simpler substances. Now stomach has gastric glands which is present throughout its wall. So gastric glands are secreting three main juices or three main elements, we can say. First is the HCl, which is the hydrochloric acid. Second, it's the mucus. Okay, and third is the two enzymes, we can say, which is inactive in nature, which is prorenin and pepsinogen. Okay. Now, let's talk about HCl first. HCl is essentially creating an acidic medium for various enzymes to act upon it. So, the most important function is creating an acidic medium in which the food can be digested or broken down into simpler substances. HCl helps in activation of these two protein digestive enzymes, or you can say by the actions of HCl, the pepsinogen in the actions of HCl, it gets converted into pepsin. Okay, we'll talk more about pepsin in some time, but let's talk about the similar thing happens about prorenin as well, by the way. So prorenin, it's getting converted into by the actions of HCl into renin. But these two enzymes have specific functions to perform. Pepsin is helping in digestion of proteins. So it's a protein digestive enzyme. Okay, it's a protein digestive enzyme. The overall reaction which happening is like proteins with the actions of pepsin with the actions of pepsin, gets converted the action of pepsin gets converted into peptides. The peptides are the smaller molecules of peptides, which are these are smaller molecules of protein which are quite simpler to digest by our human body. And when you talk about prorenin, it's essentially a milk digesting enzyme. Okay, so we even think about milk as containing lactose, and lactose is this molecule which is quite complex to digest of. So you have a special enzymes which is prorenin for digestion of milk. And if you think about the stomach containing this highly acidic, so when you think about pH is like highly acidic, okay? It's highly acidic, and hence HCl is corrosive in nature. Corrosive. So when we say that HCl is present in our stomach, definitely the inner walls of stomach could have potential threat from the strong actions of these enzymes. So, but it's protected by the actions of mucus. Mucus is like forming a layer in the inner side of our stomach, which is protecting the inner walls of small stomach from the actions of HCl. So you can see over here the blue lines which have drawn here is essentially the mucus layer, which is having the main functions to protect the inner walls of stomach from the higher acidity of the HCl. Okay, so stomach has this primary lowest converting has HCl, mucus, prorenin, propepsinogen, and it is get converting protein. So protein digestion is its main role. Afterwards, the partially digested food as such, it is entering into the intestines. Okay, now the entry or exit of food from the stomach is guarded by the stomach sphincter. The stomach sphincter is a specialized muscles which regulates the exit of food from stomach into the intestines. Now, when we say about intestines, there are primarily the two types. First, the intestines, you have the small intestines, and the one which is quite larger is called as the large intestines. Okay, and now when we say about intestines are the largest organs in our human digestive system. It is fitted into a compact space due to the extensive colon which you can see over here. The small intestine is particularly the longest of the structure which is present in our alimentary canal, but it is fitted in like probably a very small space due to the extensive coiling which is present over here. Now, the demarcation between small and large intestines is based on its diameter, not on its length. Okay, that's a point we should note. Now, intestines is essentially receiving small intestines, which is talk about small intestine. The food like from the stomach is entering into the small intestines. Now, the small intestines is receiving primarily three digestive juices. So, first is the juices from liver. Second is from pancreas, and third, this intestines itself secretes its own juices, so small intestinal juices, we can say about it. Okay, so what is when you say about liver? I will show you the liver. So liver here is a It's the largest gland in our body, and it's a brownish, red brownish in color. It's situated in the upper right side of our abdomen. Again, it's just below the diaphragm as well. Now, liver has primary function of producing bile. So, bile produced by the liver is temporarily stored in a structure called as the gallbladder. So, the one which is shown here, just behind this liver, you have the gallbladder. Gallbladder temporarily stores the bile juice produced by the liver until it is required for the action of digestion. When we say about bile, what functions bile has? So primarily, bile has two main functions. First, bile creates an alkaline medium. First, bile creates an alkaline medium for the other digestive juices to act upon. So when we say bile, it converts the acidic food which is from the stomach into alkaline medium for actions from small intestinal juices and pancreas to act upon the food particles. Next, from alkaline nature, it also has the main function of the emulsification of fats. Emulsification of fat. Emulsification is basically it's a physical process of breaking down larger sized fat globules into large numbers of fat droplets. So, for example, a larger fat molecule is shown like this, and by the actions of bile juice, it's latter converted into small droplets, which is from the actions of bile salts from the bile juice, and it's helping in increasing the surface area for the lipase enzymes from the small intestines to act upon it. Okay, this was all about bile. Next is the pancreas. Pancreas has secreting three main enzymes. The three enzymes which is secreted by pancreas include: first, lipase; second, amylase; and third is trypsin. Okay, so all of them you should just light as it's like pancreatic lipase, pancreatic amylase, and trypsin. So lipase is primarily involved in fats digestion. When you say about lipase, you are concerned about the fat digestions. Okay, helping in fat digestion or converts the fats which is present in our food into large numbers of, we can say, fats by the actions of enzymes called lipase. It gets converted it gets converted into fatty acids, large numbers of fatty acids plus glycerols. These glycerols are later absorbed by the small intestine, but you can say these are the basic in which you can convert these fats into, like in the glycerols and fatty acids. Next, you have the amylase. Amylase is helping in starch digestion. You can recall from these salivary amylase which we had in our buccal cavity. Similar function is performed by the pancreatic amylase. The pancreatic amylase helps in breaking down of larger starch molecules into its action of amylase. It's by the action of amylase it gets converted into simple sugars like the maltose and dextrine. Okay, maltose and dextrine. Next is trypsin. When we say about trypsin, trypsin is again it's a protein digestive enzyme, but the difference between the protein digestive enzyme which we have seen in stomach and the one which we are seeing in small intestine, that is trypsin, is that trypsin requires an alkaline medium for its action. So it's requiring an alkaline medium, whereas the pepsin which we have seen in the stomach, it's requiring what? It requires an acidic medium for activation. So you can see pepsin, its protein digestive enzymes helps in converting proteins into peptides, and similar function is seen in trypsin as well. Okay, now these these three enzymes are later passing on to the small intestine. So small intestine is again helping in complete digestion of this residual um, fatty acids and glycerols into one which is absorbable. Maltose and dextrine is converted into glucose. This glucose is later absorbed by the body. Peptides is converted into some usable forms which are later absorbed by the small intestine. Now, for the absorption, small intestine has a very important functions to know. Small intestine has this numerous infold structures which are present throughout its surface. This numerous finger-like projection is what we have termed as villi. These villi are present throughout the walls of small intestine, and this villi also have small macrovilli, which is present on the surface, which I'm showing in blue color. Okay, so villi has the primary function of absorbing the digested food materials, and it is richly supplied with blood vessels throughout its network. Okay, so we can say like this: they are supplied with rich amounts of blood vessels, and these blood vessels helps in absorption of food materials from the small intestine into the bloodstream. Okay, now villi also helps in absorbing as increasing the surface area for absorption, and through which the digested food material is finally entering into our blood, and through the blood it is reaching throughout the various cells of our body and utilized as per the need to perform the basic metabolic activities. Now food here is now completely digested in small intestine. We can say the food is completely digested. Complete digestion of food takes place, and last step in this process of digestion is accompanied by the large intestine. The large intestine, which is this structure, is mainly involved in absorbing the water molecules and water absorption from the undigested materials and helping in formation of feces. These feces are stored in a structure which is called as the rectum. Okay, rectum is another part of the large intestine itself, and from where the rectum, the undigested food materials or the feces are later defecated or egested out of the body via the anus. Okay, now defecation is the process in which undigested materials, we can say feces, is removed or egested out of the human body, and this marks the complete accomplishment of our complete digestion of food in our human bodies. Okay, now when we say about digestion, there is basically starch, fats, and proteins, and overall starch is digested by the amylase, fats are digested by the lipase, and the proteins are digested by the trypsines from the small intestines and pepsin from the stomach. And through this, the whole process of digestion is accomplished in the human body. Okay, so I hope that you all have understood human digestive system properly, and I will see you in the next one. Thank you.